Hello friends, welcome back to my channel. In this video, we are going to discuss MCQs on Pharmacognosy and Phytochemistry 1 Unit 3, that is plant tissue culture and edible vaccines. So let's begin. Is a method practiced for plant propagation under sterile conditions to produce clones of plants is termed as plant tissue culture. Next question is, in 1904, Dash started his research on embryogenic tissue cultivated in vitro in an artificial medium of mineral salts and sugar solution. In answer is Hanning. The next question is, Dash performed an experiment for embryo development and callus formation in Datura using coconut milk. Van Overbeek et al. The next question is, Dash in 1972 successfully obtained the first interspecific somatic hybrid by protoplast fusion of Nicotiana species. Carlson et al. The next question is, Dash is an enzyme cofactor. Manganese. The next question is, Chlorine's physiological function is required for photosynthesis. The next question is, which one is not a macro element? Iron. The next question is, which one is a micro element? Iodine. The next question is, the mixture of nitrate and ammonium ions has an advantage that the medium is weakly buffered as Dash are excreted by the uptake of NO3 negative ions. OH ions is hydroxide ions. The question is, Dash is a cheaper source of amino acid mixture and is used sometimes. Casein hydroxylase. The next question is, the most commonly preferred gelling agent is agar. The next question is, glassware used for plant tissue culture should be dipped in sodium dichromate sulfuric acid. Question is, the portion of plant body removed to establish a culture is termed as explant. The next question is, Dash is not used for surface sterilization of explant, sodium dichromate. The next question is, a medium used for production of more amount of callus is termed as proliferation medium. The next question is, subculturing of callus is repeated at an interval of 4 to 6 weeks. The next question is, Dash medium is used for inoculating the cut and surface sterilized leaf, primordia or very young leaves. Agar solidified. The next question is, Dash is the process in which radical tips of seeds germinated aseptically are cut out and cultured under controlled conditions to facilitate their growth. Root tip culture. The next question is, root tip culture are maintained in a moving liquid medium. Question is, due to limited growth potential of leaves, the amount of leaf growth in culture depends on the Dash at the time of excision. Maturity stage. The next question is, Dash is the process in which the excised floral buds are aseptically cultured in a culture vessel where they develop to produce a full bloom. Flower culture. The next question is, anthers are cultured on a Dash medium where the microspores within the cultured anther develop into the callus tissue. Nutrient. The next question is, the principle on which Dash relies is that pollen can be grown by the culture of intact anther without disturbing its natural habitat and environment. Anther culture. The next question is, Dash culture is the process involving in vitro culture of shiny dome-like structure of less than 0.1 mm length and 1 or 2 pairs of very young leaf primordia. Meristem. The next question is, in vitro ovule culture helps in determination of the factors regulating dash development to a mature embryo through organized stages. Zygote. The next question is the process of dash relies on the principle that the embryo is aseptically excised and transferred to a nutrient medium to develop under appropriate culture conditions. Embryo culture. The next question is dash are naked plant cells lacking cell wall but possessing plasma membrane and other components. Protoplast. The next question is dash originate from the eggplant infected with Agrobacterium rhizogenes. Hairy roots. The next question is which one is not a prime culture system? Embryo culture. Question is the loosely arranged parenchyma cells proliferating from mother cells form an amorphous aggregate termed callus. The next question is callus tissue show dash color if large amount of carotenoid pigments develop. Yellow. The next question is in dash phase cell division rate delays with the aging of culture. Progressive deceleration. The next question is in dash culture method system is kept open and is designed for periodic addition of fresh medium and removal of original culture. Semi-continuous suspension. The next question is the utility of plant cell biotechnology for production of pharmaceuticals has been increased by enzyme immobilization. Next question is infectious disease can be prevented by vaccines. The next question is edible vaccines are developed by dash in which the selected desirable genes are introduced into the plants and then allowed the production of encoded proteins by these altered plants. Transformation. Next question is the process of dash while maternal antibodies are present is supported by edible vaccines. Seroconversion. The next question is rabies glycoprotein is produced by tomato. The next question is Arabidopsis produces which antigen? Foot and mouth virus. The next question is dash is the base of developing edible vaccines. Genetic engineering. The next question is the desired genes are incorporated into the selected plant so that these genetically dash produce encoded proteins. Transgenic plants. The next question is which one is not a strategy employed for the production of vaccine antigen in plant tissues? The correct answer is genetic engineering. The next question is dash has gained importance in the last decade as it has many advantages over nuclear engineering. Chloroplast engineering. The next question is the genetic material is introduced into cells by transient expression method using dash as a tool. Viral vectors. The next question is in dash method the recombinant DNA is integrated into the T region of a disarmed Ti plasmid of Agrobacterium. Agrobacterium mediated gene transfer. Biolistic method is a sophisticated method which utilizes a dash to introduce the gene containing DNA coated metal particles in the plant cells. Gene gum. The next question is oral vaccination faces the problem during digestion of antigenic protein in the stomach. The next question is which one is not an advantage of edible vaccine? Vulnerable to microbial attack. The next question is which one is not a disadvantage of edible vaccine? Lack heat kill pathogens. The next question is which one is not an application of edible vaccine? Gene therapy. The next question is dash is produced in tobacco plants to treat Gaucher disease and interleukin 10 to treat Crohn's disease. Glucocerebrosidase. The next question is dash administration produces a protein present in Mousezona pellicida that inhibits egg fertilization in mice because of the antibodies formed. Tobacco mosaic virus. The next question is, at Cornell University, USA, the transgenic tomatoes were discovered to be acting against Norwalk virus. Question for this video is, potato-based edible vaccine have to be ingested raw as cooking denatures the protein and makes the vaccine ineffective. Now, thank you.